Hello everyone, and in today's lecture we are going to talk about the next water quality parameter, which is biological. So till now we have studied the physical parameters, and then we have studied the chemical parameters, and now we are heading towards the biological parameters, right? So let's move. So now we are changing our side from chemical to biological. So water quality. So in water quality, you saw we had physical parameter, and then we had chemical parameter and biological parameter. So we studied what we can observe, right? So physically and chemically. So we studied what else we can observe physically and chemically, which are present into the water. Now we will be seeing that what living organisms or biological materials are present into the water. So this is what comes under the biological parameter, right? Now the thing is, the very first thing is, what is organic matter? So organic matter in the water consists of carbon containing substances. So the matter which is having any elements which are having carbon uh, in their composition, so it will be having carbon containing substances, and originating will be the origination of them will be of living organism, waste and decomposition, right? So for example, we have proteins, carbohydrates, fats, amino acids, urea, plant residue, sewage. So plants and animals give the sewage, and then it decomposes into organic matter. So simply sewage gets decomposed into organic matter, right? So this is what the organic matter is. It's nothing but it's a waste which gets decomposed, and it's a compound which is having carbon in it. Now we have two types of organic matter, so which is biodegradable and non-biodegradable. So biodegradable, the type of organic matter is which is easily decomposable with the help of microorganisms. Microorganisms like bacteria are there. So with the help of microorganisms, pathogens and all. So if with them it's easily decomposable, we will be calling it biodegradable. And non-biodegradable is it is resistant to biological degradation. So that means bacteria or microorganisms will not be able to easily decompose them. Right now we have the biodegradable. For example, we have food waste, right? And here it is also organic matter, pesticides, but it can't be degraded easily. And then we have sewage organic matter, and then non-biodegradable in this will be having certain synthetic chemicals. And then proteins are biodegradable, some plastics are non-biodegradable. See, plastic is a polymer, and polymer contains carbon, right? So it is containing carbon. That means it's organic matter, but it will not be easily biodegradable. So it is non-biodegradable. Similarly, we have some carbohydrates, and which is biodegradable easily, right? So we will be having certain persistent organic pollutions. Now we'll see the next thing, which is why organic matter is a problem, right? And before that, we should see one more thing, that uh, if it is biodegradable, right? So it will be used by microorganisms, right? And the microorganisms will decompose it. Microorganism will decompose it, and in the decomposition it uses oxygen, so O2 will be consumed. This is very important. So for decomposition, microorganisms take oxygen, they use oxygen. So with the help of that only, they will be converting the oxygen into carbon dioxide, right? And they will be decomposing the biodegradable material, organic matter. So that means if in a certain area there is oxygen and there is organic matter, then what will happen, that organic we'll be having bacteria or microorganisms, they will attack the organic matter, and you will be seeing that if something is getting decomposed, the oxygen amount will be reduced in that. Okay, so this is a very important uh, result. When we see there is a decomposition, there will be a lack of oxygen there. So this is what the thing is. Now we have, why is organic matter a problem? So was it not good that it is there? It's okay, it's organic matter, it will decompose, what's the problem in that? So no, we can't do that, because organic matter will be getting through the microbial decomposition process, right? That means it will be consuming the oxygen. If oxygen is getting consumed, the dissolved oxygen will be reduced in water, right? And if that is reduced, that means we will be having aquatic life stress. Okay, so it is dangerous for water. If water gets below the minimum limit, then the DO into the water gets below the minimum limit, then we will be seeing that aquatic life is getting affected. So this is what, when the organic matter becomes a problem, if it is more in amount, right? More than the DO should be there, so it will become a problem. Now we are having our so if organic matter is problem, so can we say that all microorganisms in water are harmful? uh, that means till now we saw what is organic matter, why it is a problem, and what it is exactly doing into the water. Now if DO is reducing because of microorganism, then oxygen will be consumed, right? And it will be, while biodegradable material is getting degraded, right? In that means organic matter is getting degraded. So this degradation will be, the oxygen which is getting used for this, will be calling that BOD, but we will not be discussing it here. We'll be going in the treatment section, and then we will discuss that in detail, the BOD, COD, and theoretical oxygen demand and all. This is not the area, this is not the chapter that we are going to discuss that. Now now if microorganisms are present into the water, because they need to degrade the organic matter, right? They will always be there. Microorganisms are always everywhere. Even in air, we have thousands of bacteria around us. Even ICU, what is ICU? It is free from the contamination, right? But even in the ICU, you can see the bacteria are in the count of hundreds in ICU. So that's most safe and secure area for a patient, and there also we get bacteria in around 100. So microorganisms cannot be left, they will be there everywhere, in larger amount or in smaller amount, right? So the question is, are all microorganisms harmful in water specifically, because we are studying here water, right? So thus finding bacteria in water means that water is unsafe. If it is true, then so there can be two things, either it is true or it is not true. So finding bacteria in water doesn't mean it's unsafe for us always, right? So we'll be saying the answer is not necessarily, right? So not necessarily all bacteria are harmful for us, because we know that we have gut bacteria, they are in like almost half a kg of gut bacteria is there in our stomach. So if this much bacteria is there, that's a good bacteria for us, right? So not necessarily we have all the bacteria that are safe, or not necessarily we can say that all bacteria is unsafe, right? So this is what the answer is, that yeah, all microorganisms in water are not harmful. Either they are uh, and then we come to the what are indicator organisms. So the thing is that water contains microorganisms, right? And there will be microbiological water quality test, and that will often focus on the indicator organism rather than they will try to detect every pathogen. So we have in water there is a lot of type of bacteria and pathogens are there, right? Microorganisms in general. But do we test each and every pathogen? So the answer is no, we don't test each and every pathogen. We test the indicator organisms. So these indicator microorganisms are nothing but, if these are present into the water, then there may be, so these themselves are not harmful, but if they are present into the water, there may be a chance that there is harmful pathogen also present into the water. So they indicate that there can be pathogen there. So we should not have these indicator organism in water to be the water as fit for drinking or daily use purposes. So indicator organisms are the one that we are going to test if they are present or not, and they themselves are not harmful, but they will be telling us that yeah, there can be, if they are there, if the indicator organisms are there, that means there can be harmful pathogens also. Okay, so the definition is that indicator organism is a microorganism whose presence or abundance provides evidence about contamination or sanitary quality. Okay, so they themselves are not harmful, but they will tell that yeah, they came from contamination, so there can be other contamination and harmful pathogens as well. So these indicator organisms basically come from fecal contamination, and they from fecal contamination we'll be having indicator organisms, and they will tell that yeah, there is a possible presence of uh, pathogen there. Okay, so this is what indicator organism is. So these are indicators, these are not pathogens themselves, right? So this is what we should confirm. Now, what is coliform? So coliform is the one that is associated with environmental fecal contamination. So coliforms are the thing that we are going to uh, use as indicators. So these are used as indicator for sanitary quality. If these are present, that means there is contamination from like sanitary contamination, fecal contamination. If these are absent, that means water is clear. Now, uh, coliforms, why we are using that? Because it is relatively easy to detect, and we use coliforms in routine water quality testing, right? So coliforms are the indicator. So these are the indicator organisms in testing of the fecal contamination or pathogens into the uh, water quality testing. So this is what coliform is. Now we go to the question that why coliform. So we can see that pathogens can have many types, and testing each and every type will be difficult and as well as expensive, right? So in that place, if we test coliform indicator, this is first of all easy to detect, and it indicates yeah, there is a possible fecal contamination. That means we can use coliform, right? It's cheaper, it's less expensive, it's easier to test. Then why not use this? Why not spend your efforts and money and time on testing each and every pathogen, right? So we will be testing the indicator. So this is why we use coliform. Now E. coli. So E. coli is the one coliform that we use. So the full form, we can say it Escherichia, Escheri- Escherichia coli. So it Escherichia coli, and in short we call it E. coli. So the bacterium associated strongly with the intestinal tract. So if it is a fecal contamination of human or any warm-blooded animals, their E. coli is always present, right? So if E. coli is there into our sample, that means we can say yes, there is a human or animals contamination there, fecal contamination basically, and its detection is commonly used as evidence of recent fecal contamination, right? And it belongs to the broader group of coliform organisms. So fecal contamination will be tested by E. coli, and it acts as indicator for fecal contamination. Okay. Now the thing is, what is difference in coliform or E. coli? So coliform is the broad indicator group, but E. coli is a specific bacteria species. Okay, coliforms are indicator group. In this, there is a lot of type of uh, um, bacteria can be there. E. coli is a specific bacteria. So can occur in various environments strongly associated with fecal contamination. So used for sanitary quality assessment, important indicator for fecal contamination. So specifically if we are testing for fecal, then E. coli is useful. Okay. So we can't say here that all coliforms are E. coli. E. coli is one of the coliform. Okay. Now we have the microbiological testing methods. So we have three types of testing method, in which we have direct count, which is your membrane filter technique, MFT we can say, MFT. So it is membrane filter technique. Then we have SPD test, plate count specifically, and then we have MPN, membrane. So the testing will be like that. So MFT is a direct count. SPT is a colony count. We'll be discussing them in detail. And MPN doesn't count any colony or direct bacteria. So it's simply uses probability, and it gives a statistical estimation of the uh, amount of bacteria present there. Okay, so let's move to tests one by one. So the very first test we have is membrane filter technique. So in this test, what we are doing is we have the water sample, and then we'll be adding membrane filter to it. So we'll be passing the water sample to the membrane filter, and the bacteria will retain onto the membrane filter, and then we'll be placing the membrane on selective medium. And okay, so medium is given to the bacteria so that it is some it is kind of some food for that. So if bacteria will eat that food, it will grow themselves, right? So colonies will be growing around the bacteria. So if we give the selective medium to that, and we incubate it, and then colonies will develop, and then we can count the colonies and give the membrane filter value. So this is the apparatus, like this we'll be having. So we have uh, so here you can see that we have uh, sample reservoir. We have sample here. Okay, and then there is a filter funnel. You can see the filter. This is membrane filter. 0.45 micron pore size will be there for this for that membrane. And then we have clamp. It secures the funnel and reservoir together. You will be passing this water to this filter, and it will be you have this filter apparatus. And then Water sample is placed and then vacuum draws the water through the membrane, bacteria are retained, and then this filter membrane will be removed and placed to the selective medium for incubation and colony counting. So this is how we do membrane filter test. But this test is obsolete now. We don't use it that much. Okay, so we don't use it now that much, but let us see how the results are calculated in that. So let's suppose, so the results are denoted in CFU per 100 ml, colony forming unit. Okay, CFU is colony forming unit. So let's suppose that we have 100 ml of sample. We took 100 ml of sample, uh, which we filtered there, right? And then we saw that we counted the colonies. There were 42 colonies. Okay, so there were 42 colonies counted. So in this we can say that how many colonies were formed. So there were 42 CFU per 100 ml, right? So this is the unit basically. This is the unit. Okay, so CFU, and basically if I want to say like CFU per 100 ml, so this is nothing but colonies upon volume filtered, and we will be multiplying that by 100. Okay, so this is how you will be reporting the result, that is CFU per 100 ml of what sample. Okay, so this is what our results of membrane filter test are reported. Now we have the next test, which is standard plate count or standard plate count test. So in this we know that volume of the water, so we'll be taking a known volume of water, that's 100 ml or something like that, and the sample is uh, so it is incubated. The sample is incubated into a nutrient medium and then will be incubated, and then the resulting colonies will be counted. So the test is performed something like this. So we have, okay, so we have uh, the first thing, we will be having the sample. So we'll collect the water sample uh, in a sterile bottle. So everything should be sterile. We should work in like into a laminar hood, and because if there is bacteria in environment, if that comes from uh, that is there on the bottle, obviously that will also grow, and we'll count that as the colony as well, right? So that will give a wrong result. We just want what was there into our water. We don't want any contamination from environment. That's why we need everything sterile there. Okay, so we have water sample which is sterile. Now we'll be preparing the dilutions. So to prepare the dilution, what you are going to do, you will be taking 100 ml, 1 ml of this sample, and you will be adding 9 ml sterile water. So there is 1 upon total volume is 10. So it's 10, 1 upon, then it is 10 to the power minus 1 dilution. And similarly, we have and then from this we will be taking 1 ml, and then we'll be adding 9 ml. So again this became 10 ml, and 10 to the power minus 2 will be its dilution. So 1 out of 100, 1 upon 100 is its dilution, and then we'll be taking 1 ml from this and then add the water, and then its dilution will be 1 upon 1000. So simply we can say 10 to the power minus 3. So this is how we create the serial dilution. Now, so this is the serial dilution. Now what we are going to do is uh, from this from the tubes that we created, from this we'll be taking a small amount of water. uh, for example, from this tube we took zero point 0.1 or 1 ml. So basically we take 1 ml. Okay, and so we'll be adding 1 ml to this petri dish, and then we'll be adding a nutrient in this. So nutrient in this case is agar. Okay, you should remember this. So the nutrient in SPT test is used, which is agar. So agar will be about 45 to 50 degrees Celsius, and we'll be adding around 9 ml of agar, okay, to 1 ml of water. And then we'll mix that, and we'll cover that petri dish, and then we'll be putting that into incubator for 24 to 48 hours at 37 degrees Celsius. And then we see that there will be around colonies of different dilutions, and different colonies will be formed. Now you will be counting each and every colony: 1, 2, 3, 4, 5, 6, 7, 8, 9. So smaller and bigger, everything will be counted. So you should be having around 30 to 300 colonies into that. So you can get that. Now the important points are, we need to use sterile technique throughout, and choose a dilution that give, okay, so from, okay, so we'll be getting so many, so we have put so many testing the dilutions and petri dish into the incubator, right? So from out of that, some will be having the value, some will be having like only 9 or 8 colonies formed, some can have 500, 600, even 1000 of colonies formed. So we will be choosing that particular dilution which has the colony forming unit. Colony which are formed in that dilution is around in between 30 to 300. So if it is between 30 to 300, that dilution we will take that. Okay, so here, so choose the dilution that gives 30 to 300 colonies. So if too many, so colonies will overlap, it will not be accurate. Too few, then high counting error will be there. So CFU per ml will be the unit here. So number of colonies, that is around we had number of colonies, example, for example, we have 45, and then dilution factor 1 upon 100, 100, like that will be multiplied, and volume of fluid will be there. So colony, for example, here is the question, we counted 85 colonies, and then the volume we took of the sample was zero point 0.1 ml or 1 ml, and the dilution of that sample in which we counted this 85 colony was 10 to the power minus 3. So we will be multiplying all this, and we get 8.5 into 10 to the power 5 CFU per ml. So per ml, this much of colony will be formed, right? So result the report as this at 37 degrees Celsius for 28 to 48 hours. So this is what SPT test is. So it will be telling you the total viable bacterial population under the given culture condition. So you formed a bacterial culture, right? So in that, uh, we can see the CFU calculation. For example, we saw in that only, right? So we calculated it was 85. So I don't think we need to describe it again. Let's see the last test, which is most probable number, or MPN in short. So MPN is a statistical estimation. We will be performing the experiment, but we will be using probability here. Okay, so MPN is a statistical estimate of the number of viable organisms based on the patterns of positive and negative growth reaction in the replicate tube. So we'll be having positive and negative growth. So in membrane filter, you counted colonies. In your SPT test, you counted colonies there as well. But in MPN, we'll be inferring what is the population statistically. Okay, so let's see what's happening in this test. So here you can see you have uh, first we'll be taking the water sample, should be sterile. Okay, container should be closed, sterile. Now we'll be doing serial dilution, 10 to the power minus 1 dilution, minus 2, minus 3. Okay, we'll be taking from this only, right? That you should remember. You will be not taking uh, other sample. So for example, you took zero point 0.1 ml of that and then added 9 ml. So this is how it will not work. You need to take from this only, then only the, because this for this it is the set and it is subset of that, right? In probability you need to have subsets, right? So if you take from there, then the next dilution will not be a subset of this. Then how can you calculate the probability? So the dilution will be from these only. Okay, from the previous one, not the final alag sample will be there. From that, we'll be not doing that. So you will be creating three sets of every dilution. So 10 to the power minus 1, you have three sets. You can usually have 3 to 5. Okay, so of each dilution. Now you'll be adding the selective medium into that. Okay, bacteria is there, it needs food, right? So the for food in SPT we used the agar solution, but in this you'll be using lauryl tryptose broth. Okay, so lauryl tryptose broth will be used as a medium here, and then we'll be adding drum tubes. Okay, so drum tubes are a small cylindrical tubes, and we'll be adding uh, like this. It's in so the open edge of the drum tube will be at the bottom, so it will have some kind of gas into it. It will not the liquid will not go, because tube is very already very small. Test tube, very the diameter is very less. In that we'll be adding a drum tube, so that drum tube will not tilt or something. So you have to put it like this so that air will be there. Okay, so this is the main thing, that we need to have drum tube which in which air should be trapped, not the liquid that's there. Now what you are going to do, you will be incubating it for 24 to 48 hours at 35 to 37 degrees Celsius, and after that you will see in each and every test tube, you will see if the bubbles, gas bubbles are formed or not. So if gas is released, it will be showing into that drum tube. You will see in the drum tube there are little tiny bubbles there. So if the tiny bubbles are there, that means the bacteria was present and it started decomposing the uh, organic matter, and after decomposition it released carbon dioxide, and that carbon dioxide gas got trapped into the drum tube, and that we can see. So if we see bubbles, right, or gas here in this case, you see, so if we see bubbles or gas, we'll be saying that tube is positive, right? If we don't see any bubbles, that tube is negative. So the first dilution we saw all three are positive. In second dilution we saw only two positive. In third dilution we saw only one is positive. Okay, so you'll be noting the number of positive tubes in each set of dilution. So for example, I have mentioned here 10 to the power minus 3, minus 1 has 3, then this has 2, and then this has 1. So this is the combination of positive tubes. Now we'll be seeing the MPN table and we can calculate the MPN number, but I have the formula. We can calculate this MPN with the help of formula as well. Okay, so that we'll be seeing. Okay, so we have positive, positive, negative, positive, negative, negative. For like this we'll be having the test. Now let's see the numerical. Okay, how we'll be calculating that. So for that we have the formula that we should know for this is, yeah, so the formula that we should know is MPN per 100 ml is equal to nothing but positive tubes number of positive tubes upon under root ml of sample in negative tube into under root ml of sample in all tubes. Okay, so for this, let's say we have number of positive tubes are, so in this test we saw the number of positive tubes are 3, 2 and 1. So total number of positive tubes are 6. Okay, so there are 6 number of positive tubes, and then we have to divide it by the ml of sample in negative tube. So can you see the sample in negative tube, how much sample we took here? Here we took 1 ml, right? So ml of sample in negative tubes is, here there is nothing. So here there is 0.1 ml, and then plus how much is there? So 0.01 ml, right? So here the sample we took 1 by 10, right? So 1 ml was sample and total 9 ml dilution. Here we took 0.1 ml, here we took 0.01 ml, right? So 0.01, so we have 0.1 plus 0.01, 0.01 two times, so 0.02. So it came as 0.12. Okay, so ml sample in negative tube and ml of sample in all tubes. So ml of sample in all tubes is 1 ml, 1 ml, 1 ml. So we have 3 ml here, and 0.1, 0.1, 0.1 is 0.3 ml here, and 0.01, 3.33 ml. Okay, so 6 divided by under root 0.12 into under root 3.33. Okay, so right, so number of positive tubes are 6. ml of sample in negative tubes is 0.12 into total sample we used is 3.33. So if I calculate that, so I can get the answer as, so we got, wait, under root 0.12 into under root 3.33, 0.632, and then 6 divided by 0.632. So it comes out to be, the value comes out to be 9.49, and the unit will be MPN per 100 ml. So 9.49 MPN per 100 ml is the value that came from this calculation. So this is how we calculate MPN. Okay, so this is the formula. Now with this, finally we are finished with the water quality. We tested physical quality, we tested the basically we can see here a simple uh, comparison between all three tests. So filter plus count colonies, we will be growing and then count colonies, and then we'll be having statistical estimation. CFU per 100 ml, CFU per ml, and MPN per 100 ml are the results or units that will be adding. Direct colony count, yes, yes, but it's no. And then best suited for coliform, general viable bacteria, sample where direct plating is difficult. Okay, in that we'll be using MPN. So retention on membrane, colony formation, and then probability will be the principle here. So this is how MPN versus SPT versus MF works. And now we are done with all the water quality. In today's lecture, we studied biological water quality. Out of that, we got to know what's organic matter, how microorganisms work, what are indicator organisms, what is the coliform or E. coli. Then we had 3 types of tests we studied. So yeah, so this is how we come to an end of the chapter number 3, which is quality of water. Yay, we finished that. So in the next lecture, we'll be starting with treatment of water. Thank you.